Okay, welcome back. So hopefully we just had some good discussions about examples of these. So let's just really quickly review what these different kinds of selection are. So stabilizing or purifying selection is when any deviation from the norm or the status quo is going to be selected against. So you're not likely to see a lot of changes to genetic material be maintained in loci or traits that are under positives or that are under stabilizing selection or purifying selection. And this is in contrast with positive selection, where changes are actually going to be selected for, because traits at these loci are under selection. So they want to be adapting to some kind of change. And so adaptations are going to be advantageous. And so changes at these loci are going to be positively selected. So this is contrasted with something where changes are going to be selected against. These are instances where changes are going to be selected for. And this can happen in two ways. Either directional selection, which is where the optimum is now going to be it's advantageous to evolve in one particular direction. Or you can have disruptive selection or diversifying selection, where it doesn't matter in which direction that change is made so long as the change is made, because staying in the status quo is going to be selected against. And so we'll talk about specific examples of those to make that more clear. So for the examples that I came up with, so for stabilizing or purifying selection in which changes are going to be lost because they're not advantageous, so I came up with the example of human estrogen and human estrogen receptor. So if human estrogen binds to the estrogen receptor to signal for the menstrual cycle to begin, the ability of those two proteins to interact is going to be crucial for the fitness of that organism, because this is necessary for reproduction. And so the domain within the estrogen receptor that binds to the estrogen protein is going to be under purifying selection, because we don't want changes. The changes there are not going to be selected for, they're going to be selected against. So you're not likely to see a lot of changes there. So those are under purifying selection. The example that I came up with for directional selection was if we can envision a new microbe entering the human stomach. And so if it wasn't previously in the human stomach, we can assume that it was previously existing at maybe more of a neutral pH. So traits that now confer survival in lower pH, like in the human stomach, would be selected for in that microbe. Because any bacteria that are able to make a protein that allows them to live in lower pH will then survive and reproduce. And so that trait will be selected for. But we're moving in the direction of lower pH, right? As opposed to diversifying or disruptive selection, where it doesn't really matter what direction you're moving in as long as there's a change. So what's an example of this? And so classic examples of diversifying or disruptive selection are things that happen at the host-pathogen interface. So you could have come up with a lot of different examples from this one of different virulence factors and host immunity proteins that we have encountered throughout class so far. The example that I came up with was, we talked about M protein, which is a virulence factor that's encoded by group A strep. And this M protein binds to human factor H. So factor H was one of those RCA proteins that we talked about, or regulators of complement activation. So this is one of our complement proteins that actually prevents our cells from being attacked by the complement system. And so we know that group A strep makes this virulence factor called M protein that can bind factor H, and so that ends up protecting the group A strep from complement. And so humans that have mutations in factor H that prevent the M protein from binding it, but also at the same time don't prevent factor H from performing its normal functions to protect human cells, will likely be selected for, right? For example, in the case of a group A strep epidemic in a region, for example, we know that one of the most adverse effects that this bacteria can have is flesh-eating disease. So if this was a long time ago in a situation where we didn't have a lot of medical resources, the humans that encoded for a factor H that could not bind M protein would likely be selected for and have a survival advantage, right? And so so again, this isn't moving in any particular direction except from away from the ability to bind to M protein, right? And so this is a nice cartoon form explanation of what we just talked about. So this is a slide that you'll probably want to pull up on your slide skeleton, because I know a lot of these fonts are hard to see on the projector. So we'll go through them one by one. So in purifying or stabilizing selection, where a deviation from the status quo is disadvantageous in selected against, examples of host-host interactions are generally good examples of purifying selection. So for example, if this is the human estrogen receptor and this is human estrogen, and estrogen needs to be able to bind to the receptor for the signaling pathway to ensue, any mutations in the estrogen protein that make it unable to bind the receptor, or any mutations in the receptor that make it unable to bind the ligand, are going to be selected against. So this locus is going to be under purifying selection to maintain the status quo. In directional selection, we oftentimes think of this as adapting to a new environment. So if we have a protein here that's in a particular environment, blue, and then you have a change in the environment all of a sudden to red, or it doesn't have to be all of a sudden, it can also be gradual. But in the example that I gave, we're going from a neutral pH to a low pH, because that bacteria has now entered the stomach. And so any genes that confer survival in that new environment, that new lower pH environment, are going to be selected for. So you're going to get a directional selection in the direction of things that confer survival in lower pH. However, once you've now adapted to that new environment, now you're likely to be under purifying selection again. So once you've made the changes, then any deviation from the new optimum is probably going to be selected against. So we can undergo directional selection, and then once we've reached the goal, be undergoing purifying selection again. However, at the host-pathogen interface, we see a lot of examples of this diversifying or disruptive selection. And so in the example shown here, let's imagine that this is a viral protein in green, and that this is a host cell factor that's going to aid in stopping that virus from infecting a cell, or maybe killing infected cells that are harboring virus. So viruses that have mutations that disrupt the binding to the host protein are likely to be selected for, so long as the virus is still viable. Those are going to be selected for because now the virus is able to escape detection by that host protein. And so then hosts that have mutations that allow it to then again bind the virus or bind the new viral variant are then going to be selected for. So you can imagine this kind of constant one-upsmanship where the pathogen is changing and then the host is changing to try to catch up, and then the pathogen is changing again and then the host is changing again. And so we call this the arms race. And actually there's evidence of this happening in the genomic record. And so we'll talk about that. How are we doing on time? Okay, so I'm going to stop this video here and then we'll pick up again, because otherwise this segment won't end in 15. So you can start video number 3 right now.